Okay. We are now we'll move on and try to understand how the calcium is made available. Okay, but for that we need to understand how the innervation of the skeletal muscle really takes place. So I am sure most of you can understand what this. For those who cannot, let me make it very simple. We are looking at the section of a spinal cord. Okay, your spinal cord at the level of say your neck and shoulder. This is your spinal cord. Okay, and in the spinal cord you can see the H-shaped gray matter in which all the neuronal cell bodies are located, and some of the neuronal cell bodies particularly in the ventral part of the gray matter, this is a neuronal cell body. This will give rise to an axon that will come out of the spinal cord and it will go and terminate on the skeletal muscle. So any fiber that you will find, any nerve fiber that you find ending on the skeletal muscle has its cell body located where? Look at the image and tell me. It is in the on the gray matter of the spinal cord of the central nervous system. As I am talking, okay, I am controlling the muscles of my jaw, which are the skeletal muscles, which is voluntary, and I am going to control it. That's why I am able to talk coherently. That is happening because the motor cells are located in the brain. Okay, and those cells keep on sending messages to the skeletal muscle of the jaw, and then I am able to compose the sentence. Good. So where is the cell body located? In the central nervous system. It may be in the brain and the spinal cord. Any problem there? Okay, so it's a very interesting piece of information. Don't lose it. For every skeletal muscle, it has to be, look, if there is no nerve supply, the skeletal muscle is useless. The nerve supply has to be there. Okay, a nerve supply, a muscle just doesn't do anything. Okay, it needs to be told by a nerve, told by an axon, and that axon will have a cell body, a neuronal cell body, and a nucleus and cytoplasm. Everything that is located in the spinal cord, or it may be located in the brain. Okay, so far, and it will give rise to an axon that will go, and I'll call this as the innervation of the skeletal muscle. And the neurotransmitter that it releases is called as everybody should say that acetylcholine. What is it? Acetylcholine. And since it is the neurotransmitter, there is acetylcholine. Therefore, I can also say that this neuron which is sitting in the spinal cord is acetylcholinergic. What did I say? Say that loudly. Acetylcholinergic. So it's acetylcholinergic neuron, and the neurotransmitter that it will release on the. Now here we have the entire scenario. Okay, it's an amazing figure. I'll take you. Actually, we have seen all the steps, but I'll take you. I'll hold your hand and take you through. So here you have the same image. Here you have a neuron, and that neuron is innervating, and the same neuron, that neuron is innervating a skeletal muscle. Good. Now, if you stimulate this neuron. Okay, now why should that neuron be stimulated? That neuron should be stimulated because my brain decides that I want to that this is an object, and then my brain says that you need to pick up the chalk, and then my frontal cortex put the information together, and that instructs my hands. Therefore, my hands moves in the direction of the object. It picks up. Well, why it is happening? Because the information from the command that has been given by the frontal cortex has gone to the spinal cord. In the spinal cord, the signals have finally focused on that neuron which is here in my spinal cord at the level of my shoulder, and that's a cholinergic neuron, and then cholinergic neuron starts firing. Okay, and when that cholinergic neurons firing, then the muscles in my hand, they respond. How they respond? By contracting. Therefore, I am able to lift an object. So who is commanding the muscles to contract? The nerve. Okay, and how is the communication between the nerve and the muscle established? Neurotransmitter. What neurotransmitter? Acetylcholine. Good. So here you have, so this arrow, so this is so we are just focusing on this part or on this part, and we have what you call, I am sure you have done this in schools also. What do we call it as? Or motor end plate or neuromuscular junction, and that neuron I can also call as a motor neuron. What do I call it as? Motor neuron. Okay, so it's a motor cholinergic neuron sitting in the spinal cord, which sends a fiber, comes out of the spinal cord and innervates a skeletal muscle. And when this neuron fires, or when this neuron brings action potential, this neuron, this neuron at its terminal has synaptic vesicles which contain what neurotransmitter? Acetylcholine, okay. And when the action potential arrives in this part of the on the presynaptic side, on the presynaptic side, there are voltage gated calcium ion channels. The calcium will enter and it will mobilize the there will be trafficking, and the synaptic vesicles will go and merge with the plasma membrane by the process of exocytosis. Say that again. So it is by the process of exocytosis that the synaptic vesicles will release their contents, and the acetylcholine will flow in the synaptic space. So in this space you will have the acetylcholine molecule. That acetylcholine molecule will just diffuse, okay, and some of will come in contact. So what do you have? This red line here indicates the plasma membrane of what? The skeletal muscle, okay, and that plasma membrane, it has a system of proteins, okay, and that system of proteins I will call as acetylcholine receptor. What will I call it as? Acetylcholine receptor. So that's a relatively a big molecule. I will show you how big, and that molecule, that receptor molecule will have binding sites, okay, and on that binding sites will have affinity. It will have affinity for acetylcholine. Acetylcholine will combine, and once it combines, this receptor here which is sitting on the plasma membrane will respond. How will it respond? It will respond by opening. It's a channel protein. It's a channel protein. That channel protein will open, and it is selective for sodium ions. What ions? Sodium ions. Now sodium ions will go from which direction to which direction? Say that loudly. Outside. Why from outside to inside? Number 2 reasons, actually 2 reasons, because outside is more. How many times more? 14.2 more, and number 1, and number 2, they will go in because it's a polarized membrane. Outside is positive as compared to inside. Inside is about -70. So it's also the electrochemical gradient. Got my point? Electrochemical gradient which will suddenly draw the sodium ions to inside, and as a result of that, when the sodium when the sodium ions go in, originally outside was plus, inside was minus, but because of the sodium ions, something interesting has happened. What is it? Reversal of polarity, depolarization or reversal of polarity. Once then that actually that current flows. So current is flowing. Can you see this depression here? Do you remember what that depression could be about? T-tubule, you remember? Hello? T-tubule. Okay, so it goes along with the T-tubule, and as it goes, the action potential spreads along the plasma membrane and the voltage gated because of the opening of the sodium ion channels, and finally, as you go inside, you interact a very interesting kind of protein. Very interesting kind of protein, which author is showing here. That protein is actually voltage gated calcium ion channel. What did I say? Voltage gated calcium ion channel, okay, and that voltage gated calcium ion channel was in a particular conformation, and that conformation was so because outside was plus and inside was minus. As the wave of depolarization, on the arrival of the wave of depolarization, so there is a change in the electric charge. As a result of that, the protein, what protein? Voltage gated calcium ion channel will undergo a conformational change and it will open. It will open, and as the name indicates, I will tell you very quickly. What really happens is, this is interesting, get this point. This is the plasma membrane. Okay, this is the T-tube. Are you with me? Done? Somewhere in the air, okay, this is the T-tubule. Sitting on the membrane is that protein. What protein? Voltage, okay. Now that protein which is sitting here, okay, inside very close to that is endoplasmic reticulum or sarcoplasmic reticulum. Are you with me? In that sarcoplasmic reticulum, there is another receptor, another molecule, okay, another protein molecule. So what is actually happening is the two membranes, one is the plasma membrane, T-tubule is lined by plasma membrane, that membrane and the endoplasmic reticulum come very close. And there is a protein sitting here which is voltage gated calcium ion channel, and on the endoplasmic reticulum there is another protein. I will give you the name for the first time. It is called as a ryanodine receptor. Say that again. Ryanodine receptor. Again. Ryanodine receptor. You didn't say. What do you call it as? Ryanodine. Don't forget. Okay. So one protein is talking to another protein. One protein is sitting in the plasma membrane. The other is sitting in the sarcoplasmic reticulum, and they are mechanically linked. And when This guy, by this guy I mean that calcium, this thing, when this guy changes, okay, this guy also changes, because they are mechanically linked. They are mechanically linked, not electrically, mechanically linked. Actually, it is like something like one protein shaking hands with another protein molecule. And when the ryanodine receptor receives the information, it opens. And when ryanodine receptor opens, then the contents of the sarcoplasmic reticulum are now free with the cytoplasm, and the cytoplasm and the endoplasmic reticulum is the rich source for calcium ions. The calcium ion concentration, the calcium ion concentration in the sarcoplasmic reticulum may be 1000 times more, 5000 times more, I don't know, huge. So the moment it opens, the calcium ions, so here we have the, okay, I have given you one name. Now the biologists have given different names for the same thing, okay, but because textbook use it, therefore I have to tell you. I said calcium channel, hello, calcium channel. That particular calcium channel has affinity for an organic molecule called as dihydroxypyridine. Did you observe the statement? That protein molecule which is sitting in the plasma membrane, which serves as what? Which serves as calcium, which permits the, it's a calcium channel. It has affinity for what? Dihydroxypyridine. Therefore, this calcium channel is also called as DHPR, dihydroxypyridine receptor. Dihydroxypyridine receptor. What did I say? Dihydroxypyridine receptor, DHPR, very commonly used, DHPR. Okay, it's a calcium channel. It's calcium. So I'll use my newly introduced language. DHPR talks to ryanodine receptor. Okay. And when that happens, the ryanodine, and then suddenly, if I draw your attention to here is a sarcoplasmic reticulum, every red dot represents a calcium ion. Now you can see here, now it has opened, and as a result of the calcium ions have started diffusing outside. Now the calcium ions are suddenly available, and as a result of that, you can imagine the interaction between the cross sets will be excited, and then you will get the what cross bridge cycling. What will you get? Cross bridge cycling. Okay, but at the same time, you don't want the interaction to go on forever. Okay, so you want to stop it. So how do you stop it? You stop it because in the endoplasmic reticulum, in the wall of the endoplasmic reticulum, there is yet another protein system which we call as calcium pump. What do I call it as? Calcium pump. Okay, and can somebody please guess what would be the function of calcium pump? Pumping back calcium from lower concentration to higher concentration, and for that, which means work needs to be done, which means you will need energy or ATP. Okay, so this is an ATP driven system in which the calcium ions will be picked up from the cytoplasm, it will be taken, and as the calcium ion concentration drops, okay, then there is no more calcium available. So there is complete cessation of the interaction between actin myosin filaments, and the cell and the muscle cell will relax. Have we gone through the entire cycle of events? Okay, how the availability of calcium will ensure that the muscle contracts, and just read there, the skeletal muscle also known as DHPR, also known as L-type calcium channel, is a voltage gated calcium, etc. Okay, I'll move on now. Yeah.
So what regulates this calcium pump?
The question is very good. What regulates the calcium pump? The answer to that, you don't really need any regulation. It keeps on working 24/7. As long as there is calcium, and as long as the difference between the two is critical. Okay, I mean, not that every calcium ion will be picked up, that's not possible. But as long as it is within the threshold, it will keep on picking up the calcium ions from the cytosol, putting it in the lumen of the. Okay, no, there's nothing to tell that you do it now. No. If it finds a calcium ion floating around, it will pump it in. Right. Yeah, please.
What are L-type calcium channels?
Okay. Oh, good you asked. L stands for long lasting. Okay, now you can ask why long lasting. The answer to that question is, you see, as you go deeper into the calcium channels, or for that matter, as you go anywhere in biology, you find that there is not one standard protein, there are half a dozen of them. Okay. So, you have actually, if you remember, I mentioned that on the presynaptic side there are calcium channels. They are slightly different type, and we call them as P and Q type of calcium channels. Okay, the ones which I am talking about. Okay, the DHPR ones. Okay, they remain open for a little longer. Therefore, the phrase has come long lasting, and therefore we have taken that L and we start calling them as L type, which happens to be same as DHPR. Have I sorted out this thing? So, if you go into the literature, you will get even 3, 4, 5 more types of calcium channels, and they have been named accordingly. These ones are called as L type. I have also given the reason why they are called as L type. Okay, makes sense? And yeah, go ahead.
Also, the DHPR, if it's a voltage gated calcium channel, is it also ligand gated by dihydropyridine?
No. It just binds. It cannot. It's good you asked. The dihydroxypyridine cannot open the channel. It binds.
And binding improves its efficiency of.
No. Bind. No. It doesn't have the. Okay. Dihydroxypyridine is not a molecule that will operate the calcium channel. No, it just binds there. It binds there. Okay. And this binding is. You know why this binding is very important? I'll tell you why. Very interesting. Very interesting. I'll ask you a biochemical problem. I'll give you 10 grams of muscle, okay, and I'll ask you to, as a biochemistry student, please separate for me this DHPR receptor. You understood the problem? Okay, I'm giving a problem. Okay, I'll give you 10 grams. I'll isolate. I mean, it contains 100 things, 1000 things. How many? What it doesn't contain? Okay. Now I'll give you a clue. DHPR. Now use your intelligence and tell me how will you use it.
I will bind it to some agarose beads and do the column chromatography.
Something like that. Or I can use labeled dihydropyridine, okay, and then so that it will have affinity. So it will separate. Then I'll separate the rest of the things, and then maybe I'll change the pH so that the dihydropyridine separates from the molecule. Are you getting the argument now? So it binds, but that doesn't mean that it operates. No, it doesn't. It doesn't change. I mean, it's not a replacement for the molecule will respond only to the voltage changes. Got the okay.
So there's no biological function of dihydroxypyridine in the cell.
Oh no. There's no biological function. There's no okay. She's asking a very interesting question. Is there a biological function for DHPR? The answer to that question, there is no biological function. But I'll give you a clue. Can you use it as a drug? Hey, think about it. It has an affinity to modulate a channel. Are you getting argument? So such things give us to find out as to and DHPR antagonists have certain medicinal value. We'll talk about it later. You know, there's a plant. That plant is called as. Can you read for me? Rhinodine is a poisonous alkaloid found in South American plant Rhinia speciosa. Got it? And people without knowing anything about its pharmacology have been using the extract of that plant as what? Read there. As an insecticide. Now now can you, with your common sense, can you put 2 and 2 together as to why this substance was being used as an insecticide? Yeah, it is. You see, they didn't know how it works. Okay, but now we know that when you use that, it enters into the body of the, and then so this also proves that insects also have rhinodine receptors. Hello, are you getting the argument? Insects also have what? Rhinodine receptors. Okay, so this is a plant. From this plant you get an extract. From that you can isolate this alkaloid, and this you can label that alkaloid, and then you can use that alkaloid to separate rhinodine receptors from a skeletal muscle. Are you with me? And therefore that, and now you also know why rhinodine receptor is called as a rhinodine receptor. Okay, it's not because it's named after the molecule which has affinity for the protein. What is the protein? Rhinodine receptor. It has affinity for what? An alkaloid. Where do we get it from? Please read for me the name of the plant. Rhinodine. What is it? what whatever. Okay, great. So this is the ryanodine molecule that you get from here, which has affinity for that, and this is the dihydroxypyridine, which has affinity for the DHPR receptor. This is interesting. Well, a disclaimer: I don't understand much of that figure. Okay, if anybody of you can you throw light on this? This is a calcium ATPase of muscle sarcoplasm. You remember that pump we talked about? Pump. Where was it located? On the membrane of the sarcoplasm. We had a protein system. Okay. Now this is that protein system. How it really looks like. Okay. This is this crystal structure of the calcium pump, calcium ATPase, and can you see author has written here something? What does it mean? On that molecule, that's where the ATP can combine, and then it can charge the molecule, and then so this is all the cytoplasm, and this is the lumen. This is the lumen of the endoplasmic reticulum or sarcoplasmic reticulum. And your aim, whenever you want to relax the muscle, your aim is to draw all the calcium and put it back here as much as possible. And this is possible because of this huge protein molecule, which we call as simple language calcium pump. Okay, but when you say calcium pump, we generally have a very simplistic idea as but how complicated is the biology? Okay, you just to give you an overview of that, I just put this image to show how this pump is, and its function is to remove the calcium ions. And okay, this is the sequence of events you can read. This for you to write from action potential arise, depolarization of T tubules, and finally you have the calcium being pumped back into the endoplasmic reticulum for the okay. Going back to the okay, so this is the skeletal muscle. This is the skeletal muscle plasma membrane, and from here somewhere is the neuron, and the neuron has a terminal, presynaptic terminal. It releases acetylcholine, and these are and these are the where is the acetylcholine molecule? It's okay. So and in the plasma membrane you have this. What is it? Acetylcholine receptor. Please recite, say that once. Acetylcholine receptors. Are you with me? Now, the acetylcholine receptor comes into many forms. The one that you get in the plasma membrane of a skeletal tissue, that receptor, also has affinity for nicotine. You get it? Nicotine, okay, nicotine has affinity for this molecule, okay. Therefore, I can also call it as nicotinic acetylcholine receptor. Get my language? Don't forget, nicotinic, say that again, nicotinic acetylcholine receptor. So, located on every skeletal muscle, okay, at the site of neuromuscular junction, on the plasma membrane, you are going to get a protein system which will combine with acetylcholine, but acetylcholine receptors are of different types. I am not going to tell you about all that right now, but the one that is present on the skeletal muscle is called as what? Is called as, please read for me here, nicotinic acetylcholine, say that again, nicotinic acetylcholine receptor. Now please, nicotine will not function like acetylcholine. Are you getting it? No, you don't think that I can replace acetylcholine with, no, that doesn't happen. But it is actually nicotine, when it binds, it remains bind for a very long time. Okay. So when the acetylcholine or an acetylcholine like compound binds with it, this is the closed channel, and when the acetylcholine comes, the channel opens and the sodium ions will go through it, and then when the sodium ions goes through it, then the membrane will be depolarized. I just put there for your information. So okay. Just one more additional point. Nicotinic acetylcholine receptor. Say that once. It is made up of, it's a protein system made up of 5 subunits. 1, 2, 3, 4, 5. How many subunits? 5 subunits. Okay. Out of them, alpha, beta, gamma, or some of those names are called as. 2 of them have sites. Where acetylcholine can combine. And once acetylcholine combines, it opens and it will allow the. So can you see there 5 subunits there? Hello. Can you see that that cartoon there? Okay, okay, and can you see those two tiny circles where acetylcholine can combine? Once acetylcholine combines, then that protein will undergo configurational change and the channel will open and the sodium ions will go in. Now, this system is so highly conserved. What system? This system, acetylcholine, so highly conserved that whether you are a fish or a frog or a bird or a reptile or a mammal or primate or whatever, it's always a neuron in the spinal cord or in the brain that will give rise to a fiber, a cholinergic fiber. That fiber will come out of the spinal cord or the central nervous system. It will innervate the skeletal muscle, and at that motor end plate, it is always acetylcholine. It will always combine with the receptor, and the receptor is always nicotinic type. A system that is very well conserved across millions of years in the vertebrates. The neuromuscular system for the innervation of the skeletal muscle is what? Highly conserved. It is acetylcholine. And what kind of acetylcholine receptor would that be? Nicotinic. I think I'll stop now. Yes, please.
Is there any channel to remove the sodium from inside to outside?
Oh, very good question. Very good question. His question is, by this method, you will soon have the cell will be flooded with sodium ions. Cell will be flooded with sodium ions, and how do you deal with it? Sodium. The answer to that question is sitting here somewhere is the sodium potassium pump. Okay, which will keep on pumping the sodium ions to the outside, potassium to the outside in the ratio of 3 sodium to 2 potassium. Okay. Yes.
What's the role of nicotine? What does it do to the receptor?
Please ask your question again. Why does it do? Okay, I'll ask you. I will counter question. I'll get the answer from you. Where do you get the nicotine from? Drug? Oh, no, it's okay. No, wait. It's whatever the molecule. Where did the drug get it from? Plant. It's a plant. It's a tobacco plant. Come on. It's a tobacco plant. It's a plant. So the plant is making just follow through. I'll get the answer from you. The tobacco plant is synthesizing the nicotine molecule, which is abundantly present in its leaf. Why is the plant making the nicotine molecule? Why? So I'll remind you about that rhinodine. So why is the tobacco plant making nicotine? So if an insect eats it, the insect will die. So it is a protective mechanism. So we have that the mechanism which the plant has evolved to protect itself. We suddenly find that it can also stimulate the reward system in my brain, and therefore I get addicted to it. I've answered all your questions.
Yeah, but what does it do? So it inhibits the.
Oh, here. Oh no, it modulates. It modulates the action. It partly opens the channel. Okay, and after some time it desensitizes the channel. It doesn't do what acetylcholine does. It's completely different.